The cell cycle. Actively dividing eukaryotic cells pass through a series of stages known collectively as the cell cycle. The cell cycle contains two gap phases, G1 and G2, an S phase for synthesis in which the genetic material is duplicated, and an M phase in which mitosis partitions the genetic material and the cell divides. The cycle begins with G1 phase. In G1 phase, metabolic changes prepare the cell for division. At the restriction point, the cell is committed to division and moves into the S phase. In the S phase, DNA synthesis replicates the genetic material. Each chromosome now consists of two sister chromatids. The cycle then moves on to G2 phase. And in G2 phase, metabolic changes assemble the cytoplasmic materials necessary for mitosis and cytokinesis. The cycle then transitions into M phase, a nuclear division or mitosis followed by cell division or cytokinesis. The period between these divisions, such as G1, S, and G2, is known as interphase. Now, mitosis is a form of eukaryotic cell division that produces two daughter cells with the same genetic component as the parent cell. Chromosomes replicated during the S phase are divided in such a way to ensure that the daughter cell receives a copy of every chromosome. Basically, what it's doing is that it's just getting the DNA and then making a copy of it. Now, the initial stage of mitosis is known as prophase, and it is characterized by the condensation of chromosomes consisting of two chromatids, the disappearance of the nucleus and nuclear membrane, and the formation of mitotic spindle. Now, next we move on to metaphase. And metaphase, the chromosomes align themselves along the metaphase plate of the spindle apparatus. And then we transition to anaphase, which is the shortest stage of mitosis. And the centromeres divide and the sister chromatids of each chromosome are pulled apart and they move to opposite ends of the cell, and they are pulled by the spindle fibers attached to the regions. The separated sister chromatids are now referred to as daughter chromosomes, and it is the alignment and separation metaphase and anaphase that is important, ensuring that each daughter cell receives a copy of every chromosome. Now we move on to the final stage of mitosis known as telophase, and a reversal of many of the processes observed during prophase. Nuclear membrane reforms around the chromosomes grouped at either pole of the cell, and the chromosomes uncoil and become diffused, and the spindle fibers disappear. Next, you transition to cytokinesis, and this is the final cellular division to form two new cells. In plants, a cell plate forms along the line of the metaphase plate. In animals, there is a construction of the cytoplasm. The cell then enters interphase, the interval between meiotic division. Now, meiosis is the form of eukaryotic cell division that produces haploid sex cells or gametes, which contain a single copy of each chromosome, from diploid cells, which contain two copies of each chromosome. The process takes the form of one DNA replication followed by two successive nuclear and cellular divisions, known as meiosis I and meiosis II. As in mitosis, meiosis is produced by a process of DNA replication that converts each chromosome into two sister chromatids. Now, meiosis I. Meiosis I separates the pairs of homologous chromosomes. In meiosis I, a special cell division reduces a cell from a diploid to a haploid. Next up, we have prophase I. In prophase I, the homologous chromosome pairs and exchange DNA to form chromosomes. Prophase I is divided into 5 phases: leptotene, zygotene, pachytene, diploidine, and diakinesis. In the next step, prometaphase I, the spindle apparatus is formed and the chromosomes attached to the spindle fibers. At metaphase I, homologous pairs of chromosomes are arranged as a double row along the metaphase plate. It is important to keep in mind that the arrangement is just random along the plate, and that this is a source of genetic variation through random assortment, as the parental and maternal chromosomes in a homologous pair are similar but not identical. And next, we transition to anaphase. In anaphase I, the homologous chromosomes are separated and moved to opposite poles of the cells. Next, in telophase 1, the chromosome becomes diffused and the nuclear membrane reforms. In cytokinesis, or the final cellular division to form two new cells followed by meiosis 2. Meiosis 1 is a reduction division. The original diploid cell had two copies of each chromosome, but the newly formed haploid cells have one copy of each chromosome. And next, it transitions into meiosis 2, and meiosis 2 separates each chromosome into two chromatids. And in conclusion, that is how cellular reproduction takes place.